My name is Dave Champlin, and in this video, I'm going to talk through an example of how biologists use DNA to compare the relationships between any species on Earth. And we could study any species because DNA is the primary heritable molecule of all life on Earth. The example I'll describe starts with humans. And the most important job that DNA does is it has the code to code for all the proteins that each cell needs to make. Every cell needs to make the proteins it needs. It needs to make thousands of different proteins. So we're going to start with humans, and we're going to start with the human insulin protein. And the human insulin protein is synthesized in some of the cells in the pancreas. And so we zoom in on the pancreas, and these cells synthesize human insulin protein. And the human insulin protein is released into the blood, and it floats throughout the blood stream and binds to surface receptors on a variety of different tissues. The human insulin protein has a three-dimensional structure that's far too small to see even with the strongest microscope, but we know in great detail what its three-dimensional structure is. And its three-dimensional structure gives it its function. I like to use tools as an analogy of protein structure and function. This screwdriver has a three-dimensional shape that gives it its function. Insulin is like a screwdriver in a couple of different ways. It has a structure that gives it its function, and then, like a screwdriver, the insulin has different effects on the place it's used. So a screwdriver can be used to put together the engine of a car, or the seats, or the transmission of a car. And like that, the human insulin protein can be used to study how different tissues respond to the same molecule. And so muscle cells take up glucose in response to insulin binding. The liver, on the other hand, decreases glucose synthesis in response to the same molecule, insulin binding. And the fat cells make another different response. They decrease the synthesis of fats in response to that same molecule. So insulin is like a screwdriver. I like to talk that way because I grew up in a little farming town and worked a lot with tools. And then when I went on to become a molecular biologist, I found that the research labs were similar to the kind of experiences I had working in farm settings. The similarities are that everything is physical. Everything's a cause and effect relationship. You need the right tool for the right job. Everything is matter of fact. Everything has a common sense to it. And my goal in these videos is to describe that, share that with everyone. I'm trying to provide examples for folks that don't have a scientific background. I'm trying to find examples that can help folks who do research on these topics and teach these topics as well. And if you like that, please hit the like button. I make these videos to stand alone, but the title of each has a number, and they're also designed to be watched in a numbered sequence. If you like these videos, please subscribe to this channel. All right, and so the structure of insulin is well known down at its atomic level, and it looks something like this. So it has a complicated little structure that doesn't look anything like a screwdriver, but it is. It's a three-dimensional structure that confers the function on it, and that's true for every protein. Every protein has a structure and a function analogous to how every bird has a type of wings, and the shape of its wings are good for soaring, diving, and hovering, and so forth. Form follows function, always in biology, whether it's at the level of the wings of a bird or the structure of a protein. And so every protein on our planet includes this feature, and that is it's a long chain of amino acids. And so that's what's shown here for human insulin, and it's true for every protein. The little colored diamonds represent different amino acids, and it's just a long chain of these amino acids. when the cell synthesizes the protein fairly spontaneously folds up into the three-dimensional structure. So even this line of amino acids has the information for the structure and therefore the function of the protein. That's true for every protein on our planet. And the next thing to say is that we make those proteins in each one of our cells. So each cell in our body makes thousands of different proteins, and it does it by getting amino acids. Some we can synthesize, many come from our diets. So when we eat protein, we don't use the proteins in the food we eat. So if we ate a beef hamburger, there could be some insulin in there from the cow, but we would never use that protein, and that's essentially the same rule for all life on Earth, that we would break it down to the individual amino acids, and then those would go through the bloodstream to each of our cells, and each of our cells would make the proteins they need. And so the pancreas makes these proteins. And every protein is made up of similar amino acids. The difference between the proteins is the pattern of amino acids added, and that information is held in the DNA. So we're heading toward the DNA. And the DNA lines up in a line with the line of amino acids in each protein it codes. So where the line of that information matches up in the DNA is called the insulin gene. And so we're moving on to the insulin gene that codes the insulin protein. And a key concept is there'll be a linear code in the DNA that provides the information for amino acids that will fairly spontaneously fold up so that that DNA we inherit from our parents actually has the structural information to synthesize that insulin functional screwdriver-like shaped protein. All right? So once we can read that DNA letters, we can convert those letters on a computer to the predicted amino acid sequence, and this is just showing a comparison that a computer makes between different people and the amino acids they would code for insulin based on their DNA sequence letters. It's really easy to gather all the DNA sequence information from every human and compare all of their thousands of different protein coding genes like this. And we find, insulin is almost always almost identical in all humans. Very, very similar, just some little differences between people in the same way we have different hair and so forth, different hair color and stuff like that. Okay? We make those comparisons using a computer program called BLAST. So BLAST is like a Google search where we give it a word and we say, find this on the web, and it ranks the hits that it makes. And I'm just going to show a couple pages from this website. It's National Center for Biotechnology Information. I'm showing them partly because it's a publicly available data site where any person could go in and do this type of search. And so we're going to go on and we're going to first find that amino acid chain for human insulin. And the pages are too small to read in detail. That's fine. We're just going to grab a little identifier label to use to search with the human insulin. And this shows the single letter abbreviation of the entire human insulin protein. The human insulin protein is 110 amino acids long, and so that's shown down there at the bottom, and that's what we're grabbing. That's our search term, is that long chain of letters. And we just plug it into this BLAST search where we're going to plug in our little identifier label up here, so it'll go to that place and find the 110 amino acids, and then we get to pick, and I'm going to pick a domestic dog species. And so scientists have read all the DNA sequence in dogs, and we're just going to use the human insulin protein sequence based on the DNA sequence to search through all the DNA that a dog has and find the best matches and rank them. And so the computer takes 10 or 15 seconds to do that because it's a big job, and it ranks them. And that's the result. It's showing the ranking here, and this is too small to see. That's fine. The top match is the dog insulin. And there are two numbers we want to talk about in these columns because the BLAST is giving us some data. And the two numbers are, one is how similar are the dog and the human insulin protein sequences, and the answer is of the 110 amino acids that are in each, about 90% are identical. So lots and lots of similarity. We're not surprised. Humans and dogs are closely related. We're both mammals. So our genes are going to look really similar. The next thing to say is the other value. And the other value is the BLAST is measuring the probability of this match. How likelihood is this match? And it's called the expectation value, or E value. And again, a number too small to see here, but it's given, and it's given in all ranking them. It's ranking the one with the highest E value as being the best match. Now, the E value takes into consider how many genes it looked at. So how many genes does the dog have? So that's part of the probability it's calculating. So what I'm going to describe next is a probability that's essentially the same as it. There's some slight differences in it, but this will do just fine and when we make this comparison. So make this comparison. Imagine matching like this. Imagine that you take a coin, you flip it 110 times for the number of amino acids in insulin, and you keep track, heads, tails, heads, tails. And then you say, there's my list of heads and tails. Your friend now flips. Okay? You were the human, that's the human data, and your friend flips that the data is going to be what Google finds in a dog. So imagine that you match 90% of the time. More than 90 out of 110 times, you matched. Heads, 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 like that. What are the chances? Oh, it's a miracle, right? Because to match in the first flip, it's a half, just a half, 50%, half the time you'd match. To match in the first two flips, it's a half times a half, or a quarter. And it just is a half times a half times a half, and do that 90 times, like that. To match that many times is very, very unlikely. So that's what BLAST is doing, and a supercomputer is making all those comparisons. It's a big, big job that a supercomputer does very fast, because lines of information and making those line-like comparisons between species is what a computer loves to do most, okay? Here's a key concept, and it's one of the most important concepts of life on Earth, and it is weight. Because we're not talking about coins, we're talking about amino acids, and there are not two possibilities, heads and tails. There are 20 different common amino acids that are coded for by the DNA. So if the human matches with the dog at the first amino acid, and those are the single letter amino acids, so the first one matches is alanine. That's a 1 out of 20 chance. Just 5% would we expect to see it, but let's keep rolling, right? Second position, leucine. They match again, so it's 1 out of 20 times 1 out of 20, it's 1 out of 400. So we're already less than 1 in 100, and we've just gone through 2, then tryptophan and methionine, and on down the line. So the key concept and a key issue on this comparison between DNA sequence is when we look at the protein coding regions of the DNA, the probabilities get very unlikely very fast. The amino acids are 110 long for insulin. And it's 1 out of 20, 1 out of 20, 1 out of 20, and we get 90% matches. So BLAST gives us the E value for that, and the E value is based on that long sequence and the strong similarities. The chances of that matching to be that similar are very unlikely. the chance is not 1 out of a million, that would be 1 with 6 zeros, or 1 out of a billion, that would be 1 with 9 zeros, or 1 out of a trillion, that would be 1 with 12 zeros. The chance that the human are this similar to the dog is 1 out of 5, 1 out of 1 with 55 zeros. It never, ever could happen. Never, ever, 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 ever in the universe could that happen. What's it saying? What it's saying is that there's no chance that a dog was different than a human, and it ended up with an insulin protein doing that same function, right? A screwdriver binding to the surface of the muscle, increasing glucose synthesis, binding to the surface of, the dog's liver, decreasing in glucose uptake in the muscle, decreasing synthesis in the liver, and so forth. The only explanation is that those must have had a common origin and they're slightly different. They could not possibly have been started out being completely different and converged to this much similarity. It could just never happen in the universe. Consequently, I'll just look at it and talk about it this way. The human insulin and the dog insulin, we know from studies, we've studied them both, they're both screwdrivers. They do the same job in the dog and the human. Made by the pancreas, float through the blood, bind to a receptor on the surface of tissues, and cause those tissues to make those distinctive responses. It's the same function. And then we know that the structure of that screwdriver can be described in a line of letters, because the letters will fold up, and the line of letters can be converted from amino acids to the DNA letters, and we can make the DNA letter comparison and show that it's just impossible that these could have done anything other than had a common ancestral gene that diverged long ago, and as a result, the dog and human are slightly different, right? They're both screwdrivers, they just have a different colored handle, right? They're still screwdrivers, they're very, very similar. Insulin's playing an essential function, okay? So what's happening here is everyone's familiar with the idea of we can look at a dog, we can look at a human, and we can talk about it. Well, they have fur, they have a womb, they're mammals, they nurse their young, like that. Lots of comparisons in our bodies and in our proteins, and we're related, okay? We've dropped down to the gene level, and we're not talking about the organism, we're talking about the human insulin gene being so similar to the dog insulin gene. Look at the comparison, look at the number of comparisons, and it must be that there was a common ancestral gene that had identical sequences, and those have diverged a little bit over time. Yeah? We can make that same comparison with all the thousands of genes coding for proteins in a dog and human, and we see this over and over again. Okay, so the next thing we can do is we can pick on another species. So let's make a pick between human insulin and let's rank it with a fish. And this is a fish called a zebrafish that's been studied a lot. People have studied its insulin. Its insulin's doing the same job. It's distantly related from us, but fish are vertebrates, and so we're still pretty closely related to a fish. When we make the comparison, we see the percent identity has dropped down to about 50%. Because it's more distantly related from us, right? And the E value has dropped to, or increased to 1 with just 23 zeros. So not a trillion, not a billion, not a million, billion, trillion, but 1 with 23 zeros. So still, you can't come up with any other suggestion than these also must have had a common ancestral insulin gene. Again, we're dropped down to the gene level. We're not talking about fish and humans. We're talking about the human insulin gene, and that zebrafish insulin gene must have had a common ancestral gene because of the astonishing similarity between them. How far can we go? And that's a huge, huge principle in this story. Because of the astonishing similarity between protein sequences, based on the 1 out of 20 in every position, and proteins are long, so 110 positions to compare. Because of that, we have such astonishing similarity that we can go over deep, deep evolutionary time and still find astonishing similarities between proteins in very distantly related species. How far can we go? Let's give it a try. So here I took the yeast that causes bread to rise. We are distantly related to that yeast, but not very closely related to yeast that causes bread to rise. So when I did the blast search here, it came back with this statement. It said, no significant similarity. It didn't find anything like insulin in yeast. Is that because it's so distant related that we've lost all those zeros? And the answer is no. The answer is yeast doesn't have anything like insulin. Insulin is a hormone that's used in big animals to have tissues, organs respond to a signal. Yeast is a single-celled organism. We wouldn't expect to find it. We can pick other proteins that humans have that yeast wouldn't have. Hemoglobin carries oxygen to our blood cells, in our blood cells carries oxygen to our tissues. Yeast doesn't have anything like that. Yeast doesn't have hemoglobin like that, doesn't have that protein. All right, so let's pick a protein that we'd expect yeast to have and see if we can stretch. Given these incredible numbers, can we stretch all the way between humans and yeast and make that comparison? And the answer is we can. And so I picked a protein that would be expected that every cell needs to have. And the protein I picked is called CDC25, and I found the human CDC25 protein sequence on the NCBI website, and I plugged that into BLAST, and I asked it to compare it with all the genes in Saccharomyces cerevisiae, which is the genus and species that is the yeast that causes bread to rise. And it says, yeah, there's a match. And the similarity here is the percent identity is actually down, it's only 30%. So these are very distantly related. But there's still that similarity, and the chance of that happening is 1 out of, again, not a million, billion, trillion, not 6, 9, 12 zeros, but 1 out of 1 and 32 zeros. Now, wait a minute, because that chance is even lower than that zebrafish. So what's going on with that? Because the yeast are really distant related. How could we have this much similarity in such a different species than us? Is there a problem with this analysis? And the answer is no. The answer is CDC25 has 300 amino acids that we're comparing. It's a bigger protein than insulin. Insulin is a small protein. CDC25 is more similar to the typical protein size in cells, 200, 300 or larger amino acids. And so there are lots of proteins like that, and we go to the DNA sequence and make those comparisons. We can roam across all the evolutionary time of life on Earth in making those comparisons. And we can do that with thousands of species and thousands of protein coding genes. Thanks a lot.